We are now looking at proteins still, but under the heading of proteins, we said earlier that all enzymes are proteins. So now we want to know more about this structure called enzymes. First of all enzymes are proteins. They are globular proteins. What is their function? They function as biological catalysts. What is a catalyst? A catalyst is something that speeds up a reaction without itself taking part in that reaction. In other words, it does not get used up. It comes there, it does its thing, it speeds up that reaction, and it can go away and do more of what it did for that reaction. Okay? Without enzymes, biochemical reactions would occur too slowly. Why? Because the energy that we would have required to do the same reaction would be tremendously high compared to what enzyme does. So we say that enzymes reduce the activation energy. For example, let's look at a simple example. You want to boil water. When does the water boil? When the kettle makes a noise. Wrong answer. The water will boil when it reaches 100 degrees Celsius. Okay? So the activation energy for that water to boil was 100 degrees Celsius. It needed 100 degrees Celsius of heat for it to take place. Now, just like that, in our body, we have all these reactions. When glucose combines with glucose to form a disaccharide, when the disaccharide is broken down to form monosaccharides. For all these reactions to take place, you need a certain amount of energy. And what does the enzyme do? It decreases the amount of energy required for the same reaction to take place. So it makes these reactions possible. Without this, half of these reactions would not take place. Or if they did, they would have occurred very slowly, and that wouldn't have been good enough for us to sustain our lifestyles. So enzymes work rapidly, and they maintain reactions at an optimum rate. Optimum here, the word optimum is important here. Optimum doesn't mean too fast or too slow. Optimum means the best. The best at that moment. What is the best? That's the optimum. Okay? They catalyze the synthesis and breakdown of molecules. Very important statement too. So not only do they help in the breaking down of molecules, but they also help in the building up of molecules. Like I said, they can take a monosaccharide, they can help in the building of a monosaccharide plus a monosaccharide to form a disaccharide, and they can be used in that same reaction to convert a disaccharide into two monosaccharides. For example, if we take glucose and glucose, it is forming maltose. The enzyme that is involved here is known as maltase. The same enzyme maltase can also reverse this reaction. In other words, it can convert maltose to glucose and glucose. Look at the arrow. It is going both ways. So this enzyme can help in the synthesis, that way, going to the right in this reaction, or in the breaking down, going to the left in this reaction. Generally, the enzymes are named after the substrate on which they act. For example, I told you earlier, maltase works on maltose. Sucrase works on sucrose, and so on and so on. They do not, I said earlier, they do not take part themselves in the reaction. They simply lower the activation energy so this reaction can take place. They speed it up, and they move on and they carry on with what they were supposed to do. They are specific. They are specific. This is absolutely crucial. For example, maltase. Maltase can't convert sucrose. It can't work on sucrose because it only works on maltose or glucose to make maltose, or from maltose to glucose. They are specific. One enzyme will only catalyze the reaction of one particular substrate or a group of substrates. For example, the active site of the enzyme, just one small part of it, will fit only into a particular substrate. And from this, we get what we call the lock and key hypothesis, or the lock and key theory now. Let's look at this diagram. The gray blobs are the enzyme, for example, and this structure here is the substrate. Notice that the substrate, a portion of the substrate, its shape is complementary to a particular shape on this enzyme. So therefore, this substrate can fit into this enzyme as we can see there. Hence the word lock and key. I can't take any key and fit it into any car. Well, maybe these days, you know, at the rate at which the cars are getting stolen, maybe people have a master key to open the cars. They're talking about 5 minutes, 10 minutes that cars are being stolen. But no, generally, one key fits one lock. And if you try to open your back door with your front door keys, it's not going to work. This is what we are saying. Why? Because the grooves in those keys are complementary to the grooves that are found in the lock. Therefore, they fit into each other and the key can turn. Sometimes you'll find that the key fits into different locks, but it will not turn because it's not exactly the same. So it's not the key for that lock. So the same way, enzymes work on this lock and key hypothesis. Notice what happens here. Here's the substrate. Now what happens? This is a reaction in which the enzyme helps to break something down. Here are the products. The products are not the same. The products have changed from what the substrate was. Look at that. Follow that up. There was a substrate. At the end, it looks different. That will be the products. But look at the gray. 1, 2, 3. In all instances, the gray part did not change. That is your enzyme. Why? Because we said earlier, the enzyme does not participate in the reaction itself. It simply facilitates this reaction. It helps this reaction to complete itself. So that is what we talk about, the lock and key theory. We said earlier, enzymes are sensitive to temperature and pH. Enzymes function best at an optimum rate. And in temperature, that optimum rate in the human body is between 30 to 40 degrees. And the pH in different parts of the body, different enzymes have different optimum temperature, pH. For example, in the stomach, the enzymes that work in the stomach need a pH of 2, which is very low. It's a strong acid. Whereas if you go to the intestines, same food. The food is gone through the stomach into the intestine. Here the enzymes require a higher pH, 10, 11, which is actually basic, from 2 to 10, 11. Look at the difference. Look at the range of specificity of this enzyme. So in one case, a particular enzyme favored pH of 2, and in other, it may favor pH of 7, 6.5, 7, like in the mouth, and in the intestine, above 10. So in different ranges, or different enzymes have different pH range. But in terms of temperature, they all work best between 30 and 40 in the human body. If the temperature drops below 30, the enzyme activity will decrease up to a point where the enzyme becomes inactive temporarily. Remember what I said about the egg. Whereas, if the temperature rises above 40, there's a problem. Because now, the enzyme's shape will change. It's a protein after all. And its shape will change. And if its shape will change, it becomes denatured. And if it becomes denatured, it cannot work. And we can't fix it again. We can't reverse it. We can't make the shape right again. So that's where the problem is. Now, how do we use enzymes in our everyday lives? For a start, you'll be watching TV and you'll see the adverts where they talk of biological detergents. Detergents, the things that we use to clean our clothing, our utensils in the kitchen, etc., etc., etc. These biological detergents contain protein digesting enzymes, proteases, to break down stains. All this blood, sweat, gravy, egg yolk that falls on your clothing, the particular detergent would be able to dissolve those things, which normally water would not do on its own, or a non-biological detergent would not be able to do that. Enzymes are also used in the brewing industry to make beer. It is used in the dairy industry while making cheese. in different industries in the food, for example, making fruit juices. It is also used in the baking industry to make bread. These are the uses of enzymes in our everyday life. That brings us to the end of enzymes, and we now move on with vitamins or vitamins. Now, vitamins are also organic compounds. Why? Because they have carbon, hydrogen, and oxygen. And they have nitrogen. They also have sulfur and phosphorus. They're also complex organic compounds, as we've spoken earlier of the others. Vitamins generally are required in small quantities for normal growth and development in animals. We all need vitamins. Generally, however, the vitamins we need should come from the food that we eat, if we eat the right foods. Once we stop eating the right foods, then we need to now look for supplements. And you can read tons of articles, tons of research on the internet about how different vitamins taken in the wrong dosage can cause problems for you. Let's look at the vitamins that we need to know. We need to only concentrate on 5 of them. Vitamin A, B1, C, D, and E. These vitamins are given letters, and they normally refer to a group of vitamins that we need in our body. What do we need to know here? Look at the columns, headings. 1 is the name of the vitamin. 2 is where do we get it from, the food source, from which food naturally will we get this vitamin. What does this vitamin do for us? What is its function? And just like we looked at the minerals earlier, what happens if we are running short of this guy? What is the deficiency disease caused by a shortage in any one of these 5 vitamins? This is what we're going to look at. Let's start with A. Good place to start. Where do we get A from? Vitamin A. We get vitamin A from carrots. We get vitamin A from dairy products. We get it from fish liver oil. So eating fish, you're going to get vitamin A. Eating carrots and various dairy products will give you vitamin A. Why do we need it? Vitamin A is said to have an important role in eyesight, especially night vision. It also has an important role in maintaining healthy skin. Obviously, because of these functions, if we have a shortage of vitamin A, we're going to have poor night vision for a start, and our skin may become dry and hard. Remember what I said earlier on? That is related to that. The deficiency disease, it's not a difficult question. They say, predict what would happen if a person has a deficiency of vitamin A. What are they asking you? They're simply asking you the function of vitamin A in a reverse situation. In other words, what's not going to happen? Or what was it doing? If it was going to do this, if it's not there, obviously this is not going to happen. It was there for good eyesight. If you do not have it, therefore you're having poor night vision. Simple. Vitamin B1, whole grain cereals. All your cereals you have in the morning properly. Try to avoid the refined ones. Take the whole grain cereals and eat those. Those are better and more healthy. Lean meat and liver. Many of you don't eat liver, but it is a very rich source of vitamins. Function, it gives you a healthy appetite, and very important, it prevents constipation. Constipation means that your system is blocked, that you cannot go out, your bowels are not working properly. And if you do not, your system does not work, that means if your undigested food does not leave your body periodically, this causes problem. It causes discomfort. It can become toxic while this undigested food is in your body. It becomes toxic. You start getting sick. It is always important to maintain healthy bowel movement. That means you must have a healthy situation where you go very often to the toilet. And very often doesn't mean every 5 minutes. Once a day, once in 2 days for some people, and so on, depending on your cycle. But if it starts giving you discomfort and so on, then you need to go and see a doctor to find out why you're having this problem. So it prevents constipation. If you do not take in enough vitamin B1, you suffer from a condition known as beriberi, and this beriberi leads to nervous and heart disorders. It's very easy. B1, beriberi. If you have a problem with B1, you suffer from beriberi. Vitamin C, this we get mainly in our citrus fruit, lemons, oranges, tomatoes, and the green vegetables. These are things that will give us high doses of vitamin C. And look how nature works. When do we get a lot of oranges, nachis, etc., citrus fruit, and so on? In winter, when we need a lot of vitamin C. Let's see why. For a start, it prevents infection. It maintains blood vessel walls, the walls of the blood vessels. It maintains the structure of your gums, and it helps in the structure of the teeth. Vitamin C. If you do not have vitamin C enough of, you suffer from scurvy. Here you get regular bleeding under the gums and under the skin. So if you are suffering from these symptoms, bleeding gums regularly or bleeding under the skin, then you need to check your vitamin C content in your blood and take it up from there and try to increase the vitamin C that you take in. Vitamin D, dairy products, fish, liver oil, and sunlight. Interesting. Vitamin D is in your body, but without the sunlight, this vitamin D will not synthesize. So therefore, it is important in our life to have some form of sunlight so that the vitamin D can be activated with the sunlight. Sunlight is essential for the activation of vitamin D. It helps in the absorption of calcium, and it also has a role in giving us strong bones. Obviously, you can see the connection here because calcium was necessary for strong bones and teeth. If you do not have enough vitamin D, in children, they suffer from rickets, where the legs actually bend in different ways, and they can get easily fractured, etc., etc. And in adults, we have a condition known as osteomalacia. Vitamin E, we get from wheat germ, meat, and dairy products. Function, healthy red blood cells, protection against cancer, heart diseases, and also you'll see a lot of your creams talk about rich in vitamin E. It is also important for healthy skin, vitamin E. In rats, this experiment has only been done in rats, where they found that a deficiency in vitamin E leads to infertility in rats. We're not yet sure among humans. It also leads to anemia, and obviously, if you take the reverse of these things here, it can not lead to cancer, but it would not give you protection against cancer. That doesn't mean if you don't have vitamin E, you're going to get cancer. Remember, it's not a either-or situation here that we're talking about. You know, lots of children, you ask them, so what happens if you don't have vitamin E? You'll have cancer and you'll die. That's not how it works. It doesn't just go that way. Vitamin E, you'll have some problems, and these problems can lead to other problems if you do not sort them out. So it's not just a cut and dry thing that if you don't have this, this is what's going to happen. Moving on still with the organic compounds, we talk about nucleic acid or nucleic acid. They also contain carbon, hydrogen, oxygen, and P, and nitrogen. They must have nitrogen. Both the nucleic acid that you're going to study in your lifetime have nitrogen in the form of nitrogenous bases. We're not going to go into the details of that. You're going to learn more about that in grade 12. All nucleic acids consist of building blocks called nucleotides. The nucleotides are made up of sugar, phosphate, and the nitrogenous base that I spoke about. Those are the three things that you get in one nucleotide is made up of a nitrogenous base, sugar, and a phosphate. The two types of nucleic acids that you're going to come across in your life are DNA and RNA. DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. The word deoxy and the ribo come from the sugar, the type of sugar that each of these things have. The DNA has deoxyribo sugar, and the RNA has ribose sugar. We're not going to go into the details of that now because it is not within your ambit to learn that right now. The importance of these nucleic acids are, for a start, DNA carries and transmits genetic information. Genetic information is information that you inherit from your parents and you going to give to your children. In other words, the color of your eyes, the texture of your hair, the color of your hair, the size of your nose, etc., etc., etc. These things are all controlled by genes, and these genes are found on the DNA. So DNA control, or rather, is important for inheritance of characteristics. DNA and RNA together control metabolic activities of the cell. Example, protein synthesis. They control protein synthesis, and since all enzymes are proteins, and since all enzymes control metabolic activities, so therefore DNA and RNA are involved in the control of metabolic activities. Therefore, they are also referred to as master molecules. But you're going to learn a lot more about the DNA and RNA when you get to grade 12. There's a whole section on DNA and RNA and protein synthesis. Some practical work that you would need to do. We're just giving you a summary here. In later lessons, we were going to try to show you some of these practical activities live so that you can see what is actually happening. Here today, we are just going to explain what happens. For example, the first one is food tests, where we test different foods for the presence or absence of different organic compounds. For example, samples of different food can be tested for the presence of carbohydrates, fats, and proteins. So we're looking at the three main organic compounds, and we're seeing how we can test for each of them. The first one is glucose, to test for glucose. And obviously that means we want to test for carbohydrates. Not just any carbohydrate, but glucose. How do we do that? We use a chemical substance which is called an indicator, and in this case we can use fillings A and fillings B solution, or we can use Benedict solution. It's an either-or situation here. We don't have to use both. We can use either of these. In both cases, there's a color change noted if glucose is present. And this color change is from blue to orange, to that color. When the solution changes from this deep blue to the orange, then we know that glucose is present. So in other words, we can say that the color change to orange is a positive test for glucose. The next one is for starch. Starch, we use iodine solution, not just iodine, but iodine solution. And in the case of starch, the solution can change from brown, with iodine solution is normally a brownish amber color, and it changes to blue-black, a very purplish, deep purple, blue-black color. Fats, we use ether or ethanol, alcohols. What we do with this is we take the fat and we dissolve it in the ether or ethanol, and if we put a drop of that ethanol or ether or that solution onto a filter paper, If fats are present, it leaves a grease stain on the filter paper. If fat is not present, then the ether or ethanol will evaporate and you won't be able to see where that ethanol or ether was dropped on that filter paper. If you can see a spot, a greasy spot on it, a stainy spot on the filter paper, that means the substance contained fat. Proteins, we either use Millen's reagent or we can use the Biuret test with potassium or sodium hydroxide and copper sulfate. In the case of the Millen's reagent, the white precipitate, in other words, when we take, for example, egg white and we add Millen's reagent to it, it changes to brick red. In the case of the Biuret test, from blue it turns to violet. Please, when you are working in the labs with any one of these chemicals, you got to practice extreme caution because some of these chemicals are poisonous. So you got to be very careful. You don't mess about in a lab. You do what you are told. You follow instructions, and you make sure that you follow the safety precautions. When you are heating substances as well, there are certain precautions that you need to follow. Please follow your teacher's instruction, because you can lose your life or you can cause somebody else to lose their life if you do not follow these precautions. They are absolutely crucial. We can also investigate some enzyme action. For example, we can look at the enzyme catalase, where in chicken liver we use hydrogen peroxide with the chicken liver, and we see that gas bubbles are released. This is a positive indicator that there's catalase in chicken liver. It's a reaction between hydrogen peroxide and this catalase. The next one is bromelain, where we can use fresh pineapple juice and put that with a substrate called gelatin, and the solid gelatin will liquefy. This is a positive indicator for the presence of the enzyme bromelain. The last one there, proteases. Here we can use normal biological detergents, and we put the biological detergents in egg yolk, and the egg yolk dissolves. If the egg yolk dissolves, it means that the proteases in that particular biological detergent helped to dissolve the egg. We're now going to look at some questions that you may be asked for the work that we've just covered. In other words, from the beginning of organic compounds and inorganic compounds, what type of questions will you expect in an exam situation? The first question, and this is a favorite question with the examiners, they like to give you this because this is a very relevant question, because it's something that you see regularly every day. Sometimes you may not notice it. Even a packet of bread has nutritional importance of information on the packet, or your cereal, or your Milo, whatever you're having, they all have this nutritional information, and you need to read the information, not to study for life science, so that you know what is it inside those items that you're eating that you're putting into your body. Imagine you're just stuffing yourself, you don't even know what's inside there. Read the inserts to find out whether certain things are there that you're not allergic to, for example. And in this case here, they're looking at the packaging of a loaf of brown bread. They give you a list of different nutrients that are found in bread, in this particular brown bread. For example, the amount of energy in kilojoules, and the amount of different compounds in grams or milligrams, etc., going right down. And this is per 100 grams or per 80 gram serving. And the percentage of the RDA, required daily allowance, that you are supposed to take every day per 80 gram serving, approximately 2 slices, they're saying in this case here. So again, data is presented to you. In this case, the data is presented to you in the form of a table. You must be able to read the table. So before you start answering questions, you start looking at all the information that's in this table. Then you go to the questions. the first question there, list the nutrient that is required for developing muscle tissue. From here, which of the nutrients are involved in forming muscle tissue? Obviously, it is a protein. Protein from our organic compounds, the one that is involved in muscle is protein. Developing good eyesight, we spoke about that just now, that would be vitamin A. Maintaining salt-water balance, which one would that be? That would be sodium. Continue with the questions. They say, now they're taking the same table. They're still basing the questions on this table. But now, the information for these questions are not in the table. So in other words, they're using information from the table, but they're asking you questions from your knowledge. So you need to marry the two. This data that they've given you, plus the knowledge that you have in the section. You need to marry the two. So here they're saying, what is a deficiency disease if there's a lack of B1? I told you earlier, B1, berry. So the answer there is berry. And in terms of iron, when you do not have enough energy, etc., you're looking pale, you are suffering from anemia. Now here, look at this one. They're saying if a person ate 4 slices of bread for lunch, calculate the amount in grams consumed of the following. They're telling you per 80 grams serving, there's the story there. What is the amount of grams they're giving you? 80 grams is approximately 2 slices. Can you see how important it is to look at the data that's given to you? Now they're telling you he ate 4 slices of bread. How much of protein? So we go to the table, here's protein. We go across, we look at 80 grams, which is about 2 slices, and the grams there are given as 6,4 grams. Is that the answer? No. That can't be the answer because they don't want 2. They want to know in 4 slices of bread. So you will multiply that by 2, and therefore your answer is 12,8 grams. And in a normal question, if they did not say here in grams in the question, you would have got a mark for saying 12,8 and a mark for grams. In this particular question, depending on the examiner and the mark allocation, they may have given you 1 because they told you in grams already. But make it a habit, when you're answering your question, always write the units. Because even if your answer is wrong, and if they were giving a mark for unit, you're going to get the 1 mark for unit. Now, if it's 2 marks, 1 out of 2, you don't have to be a rocket scientist to know 1 out of 2 is 50%. So you're getting 1 out of 2 marks there, you're getting 50% in that particular question. Why waste it? Always make it a habit to put the unit, whether they require it or not, doesn't matter. You're not going to die by writing that gram sign there. Okay, and the next one is sugar. Carbohydrates, of which is sugar. Here's the sugar. We go here, 2,8 grams. Again, times 2 because we want to know in 4 slices of bread, 2,8, that is 5,6 grams. You must show your working as far as possible as well, because sometimes in a question like this, you would have got 1 mark for this operation that you're showing here, and then you would have got your 2 marks on the other side. So make sure that you follow that as well. Still with the table. Briefly explain why cholesterol has a 0 value. It obviously means that in this bread, there is no cholesterol. That's your first obvious answer. That if cholesterol has a 0 value in this table, it means that this bread has no cholesterol. And since bread is something that almost every person eats, it wouldn't be a wise idea to add cholesterol to the bread. Why? Because cholesterol leads to narrowing of the arteries, which can cause heart attacks. So that is why there is zero cholesterol in the bread. It's something of a staple diet. Almost every single person is going to eat bread at some time in their life. So if you're going to give everybody with their bread cholesterol, unnecessarily we're adding to their cholesterol, which can lead to a heart attack. No good. Again, interpreting graphs. Let's read what they say first. Always read the instruction first. The graph below shows the effect of temperature on enzyme activity. Independent variable, temperature. Notice in this graph, they're telling you here temperature with the unit. When you're drawing your graphs later, you'll learn that you have to give the independent variable with the unit. And if here it should have been maybe percentage or whatever unit they want to use on that side. If the unit is not given, then you can write arbitrary unit. That means imaginary units that we don't know what the unit is, but they are units that we can talk about. In this particular question, they're asking you, state the optimum temperature for this enzyme. The optimum is the best temperature at which this enzyme works. So let's look at it. Where's the point at which the enzyme activity, that's here, is the highest? And we go across the graph. You're going to use a ruler to do this. You're going to go across, and you're going to come there on that line. Here it's easy because it's a line, and that's where your thing is. You come down, and your answer is 40 degrees Celsius. Again, use the units, because here they didn't say in degrees. So 40 degrees Celsius. You may get 2 marks for that. Same question. Now they say, state the enzyme activity at 20 degrees. Now they want you to work in reverse. At 20 degrees. Here we went that way across. Now we're going to go this way. There's 20 here. At 20 we're going up. We are there. We're going to go across. Now if you look at it, it should be plus minus 1,8. You have a ruler. You can actually measure the distance between there. You can have little lines going across there as well, so you can get units in there and be as accurate as you can be. And at 30 degrees, I'm going to go up here. There's it there. Obviously, it's less than halfway, so it can't be 3,5. Because from here is 3, there is 4. So your answer has to be somewhere between 3,3 and 3,4. But like I said, if you have a ruler, you put it there, and you can make little lines to show the how many intervals are there between that and the next number, and you can give it your own units in that way as well. Still on this question, briefly explain what happened to the enzyme activity once the temperature in question 2.1 is reached. 2.1, let's go back. The optimum temperature for the enzyme. So we're going back there. So when this optimum temperature is reached, what happens? Obviously, first of all, you're going to say when the optimum temperature is reached, the enzyme activity is at its highest. When the optimum temperature is reached, that is 40 degrees, the enzyme activity is at its highest.